and other non-mammalian vertebrates. In 1984, I asked him, can you help us find the gonadotropin in lampreys? We'll collect the pituitaries for you, we'll send them to you. So with his colleagues and his students, and then subsequently with students from my laboratory, we started the search for the gonadotropin in 1984. We discovered it in 2004, so it took 20 years to determine this. We went through extensive biochemical purification. We went in the late 80s with the development of the molecular techniques. In 90s, we started looking at molecular techniques to isolate this by cloning methods. We tried every possible different way. We collected thousands and thousands of pituitaries between his laboratory and my laboratory and my students. We cloned millions and millions and millions of sequences. But it wasn't until 2004, 20 years later, that we found the gonadotropin. What was exciting was along the way, and actually by mistake, we discovered all the other pituitary hormones, like the adrenocorticotropin hormones, I need to say growth hormone. So with Professor Kawuchi and his colleagues and my students and my postdoc students, we discovered all the hormones, and we finished that in 2004 with the discovery of the lamprey gonadotropin. And so we have almost completed our work on the reproductive axis in our discovery.
In 2004, there was the discovery of gonadotropin hormone in the sea lamprey with Dr. Sauer and her collaborators. And this was a culmination of 20 years of research. The initial research involved protein purification in order to find this hormone. However, that was not successful, but as time went on, molecular techniques came to the forefront of research in which they started trying to clone this hormone, gonadotropin hormone, from the sea lamprey. And after approximately 15 years of molecular approach, in 2004, the gonadotropin was cloned from the sea lamprey. Well, the most important findings in my research in dealing with the hagfish has led to my current research, which is attempts to isolate the gonadotropin releasing hormone in hagfish, which has not been identified yet. There have been many studies that have indicated a presence of a GnRH, or gonadotropin releasing hormone-like form in the hagfish, and it was a paper that was produced with it was a paper that came out in 2005, Kavanaugh, Powell, and Sauer, in which we showed that there was potentially a cyclic nature in gonadotropin releasing hormone immunoreactivity in the hagfish. And this, in correlation with gonadal development, which was examined through histology at that time, indicated that there might be a cyclic nature into these levels and possibly a potential breeding season for the hagfish. And this is extremely important because currently hagfish, or the hag fisheries on the East Coast, is a largely unregulated industry. And it's hard to fish or harvest an animal when very little is known about its reproductive cycle and how long it would take to replenish stocks. And hopefully, if I do obtain the gonadotropin releasing hormone from the hagfish, we'll be able to apply this and learn more about the reproductive cycle on hagfish, and maybe even help the fisheries with regulations in this field.
PCR is a polymerase chain reaction in which you take a DNA template and you can amplify any section of that DNA. And to do this, you need two primers that go on either side of the amplicon, or the section of DNA you want to amplify. And you can replicate this amplifying reaction a many number of times. And when you replicate it once, so every piece of that DNA gets doubled. And then that doubled can become quadrupled, and so on. And you get this exponential amplification of your target sequence. And what I use PCR for is to clone a certain section of DNA that I'm interested in the lamprey. And it just happens to be the GnRH receptor.
My PhD project in Dr. Sauer's lab is concerned with identification and characterization of the glycoprotein hormone receptors in this animal. That is an important step in the physiology of reproduction. And at this point, I want to know how many species or types of this receptor are actually in this animal. And one way to do this is by searching or screening the genome of this animal. And you screen the genome with a DNA probe, in my case. That is, it's a piece of DNA which binds to the gene in the genome. And the genome, as you know,